Thank you to everyone here at OHBM 2023, and especially to Yara and Katerina for inviting me to this awesome symposium. Today, I'm going to tell you about how gradients of prefrontal cortex representations in the human brain support working memory. Overall, I hope you can leave with two take-home messages of how the prefrontal cortex and its neural architecture support successful working memory. First, I'll share with you how gradients of working memory representations across lateral prefrontal cortex support working memory. Then I'll share with you how individual level neuroimaging and especially neuroanatomical investigation can provide cognitive insight into working memory and broader higher level cognition. In the working memory field, there seems to be a large discrepancy between the cortical substrates for successful working memory behavior across studies and species. For example, decades of non-human primate electrophysiology studies using single unit or population recordings show that spiking or LFP activity from lateral prefrontal cortex shows that these neural activity patterns represent individual items or memoranda being held in working memory. However, these kinds of signals are much harder to detect in human lateral prefrontal cortex at the level of neuroimaging. And instead, it's much easier to detect signals associated with task rules or cognitive control. And instead, the kind of memoranda-specific or crystallized item-level representations found in non-human primate lateral prefrontal cortex are much more commonly detected in neuroimaging from human sensory cortex. While of course there's a discrepancy in resolution and neural recording method between these studies and species, there's also a large difference in the amount of behavioral training that happens before neural recordings take place. For example, orders of magnitude of tens of thousands of trials are often done in non-human primates before recordings take place. Whereas in human neuroimaging, perhaps a naive undergraduate is just explaining the rules of a task before being put in the MRI scanner. Myself and my co-authors wanted to know whether we could leverage this discrepancy into an experimental opportunity and test whether such long-term learning and behavioral training in humans can influence the kind of working memory activity patterns seen in human lateral prefrontal cortex. To do this, we turned to the longitudinal training of memory representations in a set of 3 human subjects. For each subject, we gave them 18 randomly selected, algorithmically generated fractal stimuli, which were different for each subject and balanced for things like dominant color within each image. And the first time each subject saw their set of 18 unique images was their first of many longitudinal functional MRI scans. Using a precision neuroimaging framework, each of 3 subjects was given these 18 unique fractals and completed almost every other day or weekly training on a working memory and sequence learning task, along with completing upwards of 24 2-hour functional MRI scans over the course of 4 months. In every at-home training or functional MRI scan, subjects completed a simple one-item working memory task in which they were shown a fractal image like this purple spiky fractal. And then after a jitter delay period, they had to indicate which one of 3 occluded probe images matched the item they were holding in memory. For example, here, the correct answer is the bottom right matching probe. On every trial, the fractal image presented could come from the subject's unique set of 18 fractal stimuli, or be a novel fractal that was visually similar, but changed every time the subject did this working memory task. And to ensure the highest level of data quality and robust longitudinal sampling, these were not 3 random subjects, but in fact consisted of myself and my co-authors, Ariel Tambini and Anastasia Kiyanaga. And a lot of this work has now been published in the first paper from this data set, which is now available online on Open Neuro in BIDS format. To briefly describe the full extent of this data set, here I've plotted for each subject every time the subject did a full 2-hour functional MRI scan across the course of 4 months, which includes upwards of 1,600 trials of working memory in the MRI scanner, along with 5 hours of resting state scans for each subject across the course of 4 months. And again, these data are now available for download and use on OpenNeuro and have been published in the following papers on the bottom right. The first thing we wanted to look at is, with this fun, extensive training, do we actually get better at working memory? And do we learn anything over the course of months? And in fact, we do. Our working memory behavior improves, and improves only for trials using trained stimuli. For example, on the left here in blue, both our accuracy and response time get better across the course of our first 17 sessions of functional MRI scanning, only for trials using trained stimuli and not novel stimuli. We could then leverage this learning across time to gain new insight into the cortical substrates for working memory and how they may change with extensive training. To do this, we analyzed working memory delay period activity from functional MRI activation patterns to see if these patterns of bold fMRI activity would mirror findings of item level representations more commonly found from single unit activity studies. To do this, we used a pattern similarity framework in which we could correlate single trial activity patterns from different regions in lateral prefrontal cortex, generate a similarity of these vectors of voxel activity, do this for every pairwise combination of stimuli across trials, and then split this matrix up into different categories, such as trained or novel stimuli, and allow us to test specific models and hypotheses. For example, an item level model testing whether trials of the same stimulus become more similar to each other across time than to trials of a trained stimulus to other trained stimuli. For each model we could test, we could calculate a measure of pattern strength, which tells us to what extent the empirical correlation structure in the data across trials matches the specified model that we've given. We did this for each of 6 regions across human lateral prefrontal cortex along rostral-caudal and dorsal ventral axes. And we could then ask how the selectivity of prefrontal cortex activity changes with training. And remarkably, we show that representations of item level patterns for specific memoranda emerge for trained stimuli over time. Across the course of weeks of training of fMRI sessions across the x-axis, the pattern strength on the y-axis of this item level model, which indexes item level selectivity, increases specifically in ventral mid-lateral prefrontal cortex, this green region highlighted here. And this result shows this item level identity and selectivity emerges for trained stimuli in working memory with long-term learning, and offers a bridge to prefrontal representations more commonly found in single unit activity studies from non-human primates. Because we weren't done just having fun with the working memory task, during this time we also completed a sequence learning task in which the 18 trained stimuli for each subject were divided into 2 groups, sequence and non-sequence stimuli, which we learned across the course of weeks and months were more likely to be associated with each other. We could then test whether there was a categorical boundary representation between these groups of sequence versus non-sequence stimuli. And we show that this categorical representation emerges over time, specifically in caudal prefrontal areas, as shown here in purple and yellow, showing that long-term associations transform the representational structure of delay activity patterns during working memory, even when they're not relevant for behavior during the working memory task. In sum, different types of working memory representations are detected along different areas along a rostral-caudal axis along lateral prefrontal cortex, and even more changes beyond these representational ones I mentioned can be found in our paper here on the bottom right from 2022. We show that the role of lateral prefrontal cortex shifts as these stimuli become well learned and embedded in associative structures, and that these results hold important indications for bridging models for working memory maintenance from non-human primate electrophysiology to human fMRI. But not all questions are necessarily suited to this kind of longitudinal small subject scanning. And another goal of my research is to help us leverage the insights from this precision neuroimaging framework to a wide array of studies to more broadly understand the organization of human prefrontal cortex that supports working memory and other high-level cognition. For example, on this graph of subjects on the y-axis versus hours of data, how can we leverage the lessons of precision neuroimaging and apply them to data sets and questions which are only applicable, for example, individual variation in cognition to hundreds or thousands of subjects? Again, with the idea of trying to map something like a cognitive globe in human prefrontal cortex. One reason this is so hard to do is that anatomical structures in human lateral prefrontal cortex that we often study using template or average brains, shown on the bottom row, are missing and not representative of what can be detected within individual brains, shown on the top row here. These structures, called tertiary sulci, are not irrelevant, but rather can be used to help us map this cognitive globe of functioning across human prefrontal cortex in a myriad of multimodal data sets and questions. I'll highlight two specific instances in which we've leveraged this approach of individual level neuroanatomy to make broader inferences into individual variation in cognition. For example, identifying small structures within individuals that are missing from template brains and quantifying their variation across development within individuals can show that their morphology in prefrontal cortex predicts individual differences in behavioral development for both reasoning tasks and even for working memory performance. Again, when studies of individual variation reach the level of hundreds or thousands of subjects, such excruciating neuroanatomical labeling is not feasible, and certainly I don't want you to do that. So another thing we're working on and have shared is the ability to use both algorithmically generated from deep learning or manual segmentation labels of these tertiary sulcal structures that can be applied to make predictions about their location in unseen brains. Overall, I hope I've shown you how gradients of working memory representations across prefrontal cortex support working memory behavior, and how individual level anatomical investigation of human prefrontal cortex, leveraging the lessons and tools of individual level functional and anatomical neuroimaging, can help us make broader insights into brain behavior relationships in working memory and for higher level cognition in general. So I'd like to thank my graduate school mentors, Mark Esposito and Kevin Weiner, who I did most of this work while I was in their labs, and my current postdoc mentors, John Murray and Amy Arnston. Please let me know if you have any questions, and I look forward to seeing the rest of the symposium. Thank you so much.